Good day, everyone. So we are down to the last part of the nucleic acid report, which is about the anticodons, tRNA molecules, and translation. So today we are diving into one of the most fascinating processes that keeps life running at the cellular level, the translation. So I will explain how tRNA, tiny but mighty molecules, and their anticodons help decode our genetic instructions to build proteins, which is the building blocks of life. So by the end of this video, you will see just how this molecular teamwork drives everything from muscle growth to enzymes in our body. But before that, I am going to introduce myself first. My name is Gujelde, and I am from BSN 1A. So let's jump in. The amino acids used in protein synthesis do not directly interact with the codons of an mRNA molecule or a messenger ribonucleic acid molecule. Instead, transfer ribonucleic acid molecules or the tRNA molecules function as intermediaries that deliver amino acids to the mRNA. At least one type of tRNA molecule exists for each of the 20 amino acids found in proteins. All transfer ribonucleic acid molecules have the same general shape, and this shape is crucial to how they function. So the general two-dimensional clover leaf shape of a tRNA molecule is a shape produced by the molecules folding and twisting into regions of parallel strands and regions of hairpin loops. The two features of the transfer ribonucleic acid structure are of particular importance. The first importance is the 3' end of the open part of the clover leaf structure is where an amino acid becomes covalently bonded to the tRNA molecule through an ester band. Each of the different tRNA molecules is specifically recognized by an amino acyl tRNA synthetase enzyme. So these enzymes also recognize the one kind of amino acid that belongs with a particular tRNA and facilitates its bonding to the tRNA. So the second importance is that the loop opposite the open end of the clover leaf is the site for a sequence of the three bases called an anticodon. So the anticodon is a three nucleotide sequence on a tRNA molecule that is complementary to a codon on an mRNA molecule. So this is a picture or an illustration of a tRNA molecule or a transfer ribonucleic acid molecule. So the amino acid attachment site is at the open end of the clover leaf or the 3' end. So I'm going to circle the 3' end or the amino acid attachment site in a tRNA. So that's the amino acid attachment site. And the anticodon is located in the hairpin loop or in the opposite of the open end. I'm going to circle the anticodon. So here's the anticodon. So, and that amino acid attachment site and the anticodon is the two important part of the tRNA structure. So we're done with the structure of the tRNA, and now let's find out what are the functions of tRNA. So first, the amino acids attach to the 3' end via ester bonds, or the amino acids attach to the open loop of the tRNA. And second is that the anticodon loop binds to mRNA codons through complementary base pairing. So the tRNA activation is facilitated by amino acid tRNA synthetase enzymes. So the interaction between the anticodon of the tRNA and the codon of mRNA leads to the proper placement of an amino acid into a growing peptide chain during protein synthesis. So this interaction, which involves complementary base pairing. An amino acid tRNA synthetase, or this one, it has an active site for tRNA. This is the active site for tRNA, and it also have a binding site for the particular amino acid that is to be attached to that tRNA. So this is the active site for the amino acid that will be attached to the tRNA. So the interaction between the anticodon of the tRNA and the codon of mRNA, which involves complementary base pairing, governs the proper placement of amino acids in a protein. So we are done about the structures and functions of tRNA. And now let's move forward to determining codon and anticodon relationships. So a tRNA molecule has the anticodon of 5' and AAG 3' N, with which mRNA codon will these anticodon interact. So the interaction between a codon and an anticodon has directionality considerations, or it is antiparallel. So if the anticodon starts from 3' N, the codon will start with 5' N. So the given anticodon with reverse directionality is 3' N GAA 5' N. And its codon interaction, which involves complementary base pairing, is the anticodon is the 3' N GAA 5' N. So the codon for that is 5' N C U U and 3' N. So the complement for G in the anticodon is C, and the complement of A in the codon is U. So the codon is 5' and CUU 3' N. So in the last slide, we determined the codon and the anticodon relationship. And now let's determine the anticodon tRNA amino acid relationship. So a tRNA molecule possesses the anticodon of the prime and a 5' and CGU and 3' N. Which amino acid will this tRNA molecule carry? So the codons, rather than anticodons, are involved in the genetic code relationship. So let's remember that. Again, the codon is the one that is involved in the genetic code relationship, not the anticodon. So the codon, with which the anticodon 5' and CGU 3' and base pairs is 5' and ACG 3' N. So remember, as shown in the last slide, that the codon anticodon pairing involves the anticodon 3' and UGC 5' N, the anticodon written in the 3' N to 5' N direction. So again, the anticodon should be written in 3' N to 5' N direction. So the anticodon 3' and UGC 5' N have the complement codon of 5' N ACG 3' N. So the amino acid that the codon ACG codes for is 3-unit. So when we are going to find out what is the amino acid For that certain codon, we need to refer to the genetic code table, which is the other reporter already talked about. So we're just going to refer to that table. So we are done with the tRNA, their functions, their structure, and the relationship of the codon and the anticodon, and the relationship of anticodon and the amino acid. And now let's move on to the translation. So translation is the process by which the messenger ribonucleic acid, the codons, are deciphered and a particular protein molecule is synthesized. So the substances needed for the translation phase of protein synthesis are mRNA, tRNA, amino acids, ribosomes, and a number of different enzymes. So a ribosome is an rRNA protein complex that serves as the site for the translation phase of protein synthesis. The number of ribosomes present in a cell for higher organisms varies from hundreds of thousands to even a few million. So the recent research concerning ribosome structure suggests that the following for such structures. So first is that they contain four rRNA molecules and about 80 proteins that are packed into two rRNA protein subunits, one small subunit and one large subunit. So the second is that each subunit contains approximately 65% rRNA and 35% protein by mass. So third is that a ribosome's active site, the location where proteins are synthesized by one at a time, addition of amino acids to growing peptide chain, is located in the large ribosomal unit. So the fourth is that the active site is mostly rRNA with only one of the ribosome's many protein components being present. So because rRNA is predominant at the active site, the ribosome is thought to be an RNA enzyme that is a ribozyme. So the mRNA involved in the translation phase of protein synthesis binds to the small unit of the ribosome. So again, the ribosome have two ribosomal subunits. So the first one is the large subunit, which is an active site for protein synthesis, and the second one is the small unit, whereas it is the location where the mRNA involved in the translation phase of protein synthesis binds in this location. So we are done with the structure of the ribosome, the two subunit of ribosomes. Now let's move forward to translation. So there are five general steps to the translation process. The first one is the activation of tRNA. The second is initiation. The third is elongation. The fourth is termination. And the last one is the post-translation processing. So this diagram shows the flow chart of the translation process. So now let's talk about the first step of the translation process, which is the activation of the tRNA. So there are two steps involved in tRNA activation. So first, an amino acid interacts with an activator molecule or an ATP to form a highly energetic complex. So this complex then reacts with the appropriate tRNA molecule to produce an activated tRNA molecule. So a tRNA molecule that has an amino acid covalently bonded to it at its 3' end through an ester linkage. So this is a diagram or a picture of an activated tRNA. So this one also is the activated tRNA with the highly energetic complex amino acid with the ester linkage. So that is the activated tRNA. So again, activated tRNA um, is formed when the amino acid that is highly energetic complex is attached to the accurate or to the accurate tRNA. So again, I'm going to repeat the process of the initiation. First, the mRNA binds to the small ribosomal subunit. The second is that the first codon or the AUG is or will align to the P site or the peptidyl site. The third one is that the corresponding tRNA or the anticodon UAC methionine binds to AUG. So, and lastly, to complete the process, the large ribosomal subunit attaches to complete the initiation complex. So next step is the elongation. So, or the step three is elongation. The next to the P side or the peptidyl site in an mRNA ribosome complex, the second binding site is called the A site or the amino acyl site. So at this second site, the next mRNA codon is exposed, and a tRNA with the appropriate anticodon binds to it. So with amino acids in place at both the P and the A sites or the peptidyl sites and the amino acyl site, the enzyme peptidyl transferase, again, the enzyme peptidyl transferase affects the linking of the P site amino acid to the A site amino acid, forming a dipeptide. So such peptide bond formation leaves the tRNA at the P site empty and the tRNA at the A site bearing the dipeptide. So the empty tRNA at the P site now leaves that site and is free to pick up another molecule or another molecule of its specific amino acid. So simultaneously with the release of the tRNA from the P site, the ribosome shifts along the mRNA. So this shift puts the newly formed dipeptide at the P site, and the third codon of mRNA is now available at site A. So to accept a tRNA molecule whose anticodon complements this codon, the movement of a ribosome along an mRNA molecule or the movement of a ribosome along an mRNA molecule is called translocation. So translocation is the part of translation in which a ribosome moves down an mRNA molecule 3 base positions so that a new codon can occupy the ribosomal A site. So now a repetitious process or a repetitive process begins. The third codon now at the A site accepts an incoming tRNA with its accompanying amino acid. Then the entire dipeptide at the P site is transferred and bonded to the A site amino acid to give a tripeptide. The empty tRNA at the P site is released. The ribosome shifts along the mRNA and the process continues. So the transfer of the growing peptide chains from the P site to the A site is an example of acyl transfer reaction. So again, elongation is a site an elongation is a process which is the A site or the amino acyl site accepts the next tRNA molecule with the appropriate amino acid. And then the peptidyl transferase links amino acids at the P and A sites. Again, the enzyme peptidyl transferase links amino acids at the P and A sites. And then the ribosomes moves, the movement is called translocation, shifting the mRNA to expose the next codon. Now we are down to the second to the last part of the translation process, which is the termination. So in this process, the polypeptide continue to grow by way of translocation or the movement of ribosome until all necessary amino acids are in place and bonded to each other. So the appearance in the mRNA codon sequence of one of the three stop codons or the UAA, the UAG, and the UGA terminates the process. Again, the three stop codons are UAA, UAG, and UGA. And this stop codons, when it appears in the mRNA, the process will be terminated. So no tRNA has an anticodon that can base pair with these stop codons. The polypeptide is then cleaved from the tRNA through hydrolysis. And finally, we're now at the last process or the last step of translation process, which is the post-translation processing. So some modification of proteins usually occurs after translation. So this post-translation processing gives the protein the final form. So it needs to be fully functional. Some of the aspects of post-translation processing are the following. First, in most proteins, the methionine or the meth residue that initiated protein synthesis is removed by a specialized enzyme in a hydrolysis reaction. A second hydrolysis reaction releases the polypeptide chain from its tRNA carrier. So second, some covalent modification of a protein can occur, such as the formation of disulfide bridges between cysteine residues. So again, the post-translation processing step involves removal of the initial methionine residue, the formation of disulfide bridges between cysteine residues, and lastly, the protein folding into its final conformation. So finally, we are done with the translation process. So now let's sum up or let's summarize the translation process. So here's the key points. So first is that the tRNA molecules connect amino acids to mRNA codons. Second, ribosomes synthesize proteins by linking amino acids step by step. And lastly, last key point is that translation has five main phases, which are the first is the activation, second is the initiation, third is elongation, fourth is termination, and lastly is the post-translation processing. And that concludes my report about tRNA anticodon molecule and the translation process. And also that ends the report of our group, which is the group 5 about nucleic acid. So thank you so much for watching, and I hope you gain a lot of lesson all throughout the discussion. So enjoy the holiday season. Bye-bye!